Chapter 25, Metabolism and Energetics. Metabolism is the sum of all chemical reactions occurring in an organism. The functions for metabolism are to do metabolic turnover, which is the periodic breakdown and replacement of the organic components of the cells, for growth and cell division, and also for various special processes such as secretions, muscle contraction, propagation of an action potential, which is the electrical impulse which your nervous system does. Now, metabolism, it's all the reactions. So some of these reactions will be breaking down molecules. Some of these reactions will be building up molecules. So we break into two subcategories. The first is catabolism, and this is the breakdown of organic substances. And when it breaks those substances down, those substances will have energy stored in their bonds, and we're breaking the bonds so that energy will be released and be used to make ATP. We break triglycerides down to glycerol and fatty acids, proteins to amino acids, and carbohydrates to short carbon chains. We'll take that energy and we'll take ADP, use that energy to add that third phosphate on and make ATP. The second method is anabolism, and this is synthesis of organic molecules. This means we're going to take small molecules, put them together, and build a larger molecule, where catabolism was taking a large molecule, breaking it to smaller. With anabolism, we are going to use ATP, whereas with catabolism, we made ATP. We need to do an anabolism to synthesize organic molecules for maintenance and repair, support and growth, to produce secretions, build nutrient reserves, such as when we make the triglycerides through lipogenesis, or we make the glycogen, which is the storage form of glucose, through glycogenesis. Oxidation-reduction reactions, also called redox reactions, are coupled reactions. Oxidation is the removal of an electron from a molecule, and reduction is the addition of the electron. These two have to be coupled together. So when we take the electron from one molecule, we have to give it to another molecule. We don't just throw it away. Now, what you want to think of as this electron is that it really symbolizes energy. So when I take an electron from one molecule and give it to the other molecule, they're now holding the energy. Now they have two choices. They can take that energy and they can pass it on to another molecule. Therefore, they're doing more redox reactions, or they can actually use it to do something with it. Often we'll see a number of redox reactions in a chain form. So when one molecule removes the electron, it gets oxidized. The next one receives it, it's reduced. Then when molecule number 2 removes the electron, it is now oxidized, and molecule 3 that it gives it to is reduced. Now, electrons are extremely small, so it's hard to pass this along. Just think of it if you were passing along like a tiny ring, and instead of just passing it as the ring, you put it in a little box so you don't lose the ring, and it's easier to see the actual box and you don't lose it. We will often pass the electron in the form of a hydrogen. A hydrogen has the atomic number of 1, which means it has 1 proton and only 1 electron. So I can give the molecule a hydrogen, and what I'm really doing is giving them the electron, but I'm giving them in the box, the hydrogen box. The reactions where you pass it along as a hydrogen are called dehydrogenation reactions. So if I have a molecule whose name is NAD, and then I give him the hydrogen, now he becomes NADH, and he's holding the energy. When he hands that hydrogen to another molecule, it goes back to being NAD, he's lost the energy. Catabolism releases this energy via the redox reactions. Okay, so we're going to do a number of catabolic reactions to release that stored energy, and it is going to be all these redox or oxidation reductions, and then we're going to use that energy that we released to make ATP. ATP production is called phosphorylation. Phosphorylation is the addition of a phosphate molecule to ADP, the diphosphate, to make it into ATP, triphosphate. There are two ways of doing phosphorylation. There is substrate level phosphorylation, where a high energy phosphate is directly transferred from the organic molecule to ADP and then to ATP. Okay. That means that we just immediately walk over to ADP and we give them that phosphate, and that's it. There's just two players involved in that. Oxidative phosphorylation are redox reactions. Remember, we just did those, where we're going to pass that energy from one molecule to the other in the form of an electron or hydrogen. Then we'll pass it to the next molecule, and we'll have a whole chain of molecules that we're just passing the energy down. And finally, the last molecule that holds the energy decides to do something with it, and that is to make ATP. So the molecules we're passing it to, the electron to, are called cofactors or carrier molecules. And these are NADH and FADH2 when they are holding the electron. NAD and FAD when they don't have the electron. The electrons are then passed through these carriers in what's called the electron transport chain, and that will ultimately release the energy that is needed to make the ATP. What we are going to do to make ATP is break down carbohydrates. So we're doing carbohydrate metabolism, and the carbohydrate we're going to use is glucose, C6H12O6. This is a process that requires oxygen, so we're going to add the 6 O2s on one side of this equation. After we are completely done, we'll have converted those 6 O2s into 6 CO2s, carbon dioxide, which is waste. We'll have made 6 waters, H2Os, which is considered waste product, and we have released all the trapped energy that was trapped in those bonds of the C6H12O6 and produced 36 ATP. Now this whole reaction is going to require a number of different cycles, and each cycle will have a number of steps because we have to do baby steps. We have to slowly rearrange the different molecules of the chemicals so that we can release a little energy at a time without burning up the cells. So these are the bullets for the first step, and the first step is called glycolysis, which means glucose breaking. We're starting with glucose, the 6 carbon molecule. This process is going to occur in the cytoplasm, and it is an anaerobic process, which means it doesn't require any oxygen. Those two characteristics mean that every living organism can do glycolysis. Okay? You can be a little bacteria that doesn't use oxygen and doesn't have any organelles, just has its cytoplasm, and it can do glycolysis. This is a 7-step pathway. I'm just telling you that so you understand it's baby steps. We're going to start with glucose, the 6 carbon molecule, and we're going to produce at the end 2 pyruvic acids, also known as pyruvate, and these are each a 3 carbon molecule. So we've lost no carbons. Started with 6, now we have 2 times 3, so we still have 6 carbon molecules. To start the process, we're going to use a couple ATPs. Okay, think of this sort of like if you were doing a lemonade stand. You'd have to put money into it to buy the lemons and the cups and the sugar to then hopefully by the end of the day have gotten your money back you put out and plus made some more money. So we're going to use the 2 ATPs just to get the process started. And then during those 7 steps, we're going to make a total of 4 ATPs. And this is all by the substrate level phosphorylation, where we just hand the phosphate right to the ADP to make ATP. And that's going to allow us, if we used up 2, we made 4, that we have a net gain of 2 ATPs, which we can use right now, okay, and burn them if we need to. We're going to think of those as cash. So I have 2 ATPs or $2. I can go get a cup of coffee right this minute. I'm also going to make 2 NADHs. Remember, these are molecules that will store the energy. They're the cofactors or electron carriers. And they're going to store the energy, and we're going to eventually get the energy out, but not until the 3rd cycle, which is called the electron transport chain. So going along with the concept that the ATP is equal to cash, we're going to consider the NADHs and any FADH2s we make later as checks. So we're just going to put them aside, and we're going to save them, and then later we're going to go to the bank so we can cash them and convert them to ATPs or to cash. That bank will be called the electron transport chain.
Like that happen. You take
Okay. When you get to the end of glycolysis, the pyruvic acid can either enter into fermentation if there is no oxygen available, or it can go into cellular respiration if oxygen is available. Now, no oxygen might not be available because you're an anaerobic organism, or just because right at that moment there isn't any oxygen making its way to the cells. In fermentation, the pyruvic acid will be broken down a little bit more, but it does not produce any more ATP. You will recycle the NADH back to NAD so it can go back up to glycolysis and be reused. Okay. You are going to require an organic molecule to be the final electron acceptor. That's different than in cellular respiration. And if it's an organic molecule, that means you're leaving a lot of stored energy behind. So we're not getting all the energy out that we can or that we will get out of the molecules when we do it in cellular respiration. In humans, we will do fermentation and we will produce lactic acid as a waste product. We do fermentation whenever our muscles contract and squeeze the blood vessels and stop the blood from flowing. The cells still need oxygen. There isn't any right then, and they still need to make ATP, so they quickly turn to fermentation and produce it. Normally it's not a problem because the next second the blood vessels are back pumping again and it's fine. However, if you exert yourself too much, say you run a marathon, by the end of that marathon you have run out of oxygen to make ATP, so you have resorted to doing fermentation for ATP production, and you produce a large amount of lactic acid, and that can cause you to be in severe pain and cause problems with the cells. If oxygen is available, the pyruvic acid is going into cellular respiration. Cellular respiration requires that the final electron acceptor be an inorganic molecule, and that will be oxygen. ATP is going to be created through oxidative phosphorylation, and this will include 3 different steps: the preparation for the citric acid cycle, the citric acid cycle itself, and the electron transport chain. In this diagram, we're going to look at the preparation for the citric acid cycle, which is just one step, and then the citric acid cycle itself. Now, don't get overwhelmed by these diagrams. I'm not going to ask you all the chemicals involved or expect you to have everything memorized, but you have to understand the concepts. Definitely know the bullets that are here. So when we look at the prep for citric acid cycle, which is the top step there, going from pyruvate to acetyl-CoA, okay, this is going to occur in the mitochondrial matrix. Now remember, glycolysis was in the cytoplasm, so now we've moved into the mitochondria. The matrix is the middle of the mitochondria. Recall that the mitochondria is a double membrane organelle, and in the very center of the innermost membrane is the matrix, and this is where you'll find the enzymes for the citric acid cycle. It is an aerobic process. It will use oxygen, and from here on out it's always going to be aerobic. The cycle is going to occur twice for every single glucose molecule, because if you recall in the beginning, we had 1 glucose molecule and we made it into 2 pyruvic acids from that 1 glucose molecule. So now each of those pyruvic acids must go through the prep for the citric acid cycle and then through the citric acid cycle. Okay. At the end of the prep, we're going to produce acetyl-CoA. This is a 2-carbon molecule, which means that we started with the 3-carbon pyruvate and we have a 2-carbon acetyl. So we lost a carbon and formed carbon dioxide, the waste product. And we have taken that energy that we removed when we pulled that carbon off, and we have put it with NAD and made NADH. Recall that's the check. We're going to put that over on the counter. We're going to leave it for now, and then we'll cash it in later. Okay. And looking at the summary, we started with 2 pyruvic acids. We produced 2 acetyl-CoAs, 2 carbon dioxides, and 2 NADHs. The CoA is going to be released from the acetyl group, and that will be recycled back in to the prep, and the acetyl group will enter into the citric acid cycle. A new term here, decarboxylation. This is the process of taking carbon and oxygen and making carbon dioxide. Now we're going to enter into the citric acid cycle. This is a much bigger cycle than the prep. It is 8 steps, also aerobic, also in the mitochondrial matrix. The function of this cycle is to remove all the hydrogen atoms all those atoms from the organic molecules and transfer them to the coenzymes or carrier molecules, NADH and FAD, to make them into NADH and FADH2, therefore giving them the energy. The excess carbon and oxygen will be given off as carbon dioxide. When it comes in, this 2-carbon acetyl binds with a 4-carbon oxaloacetic acid to form citric acid. That's why it's called the citric acid cycle. Citric acid is a 6-carbon molecule. We're going to take that 6-carbon citric acid and break it back down to the 4-carbon oxaloacetic acid, and it's going to, in those 8 steps. Okay? And then it can start over again. The cycle is going to occur 2 times for each glucose, and it is going to produce from both cycles 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs from that 1 glucose molecule. So at this point, between the prep and the citric acid cycle, we have broken down all 6 carbons to 6 carbon dioxides. So we're done breaking it down. We've taken all the hydrogens, removed them, and added them to NAD, making NADH, and FAD, making it FADH2. And we've made 2 more ATPs. So going back to our cash concept, we have 2 more ATPs, $2 more. We can buy another cup of coffee. And then we can go to the bank and take all of our checks, NADH and FADH2s, to the bank. Remember, we made 2 ATPs in glycolysis also, in addition to the NADH. So the summary for the citric acid, we sent in 2 acetyl molecules, 2 carbon molecules. In the end, we made 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs. So we have completely broken glucose down at this point. We can throw out the carbons as carbon dioxide. We got rid of some of the other hydrogen, oxygen as water, H2O. And we now have what's left to do is to harvest that energy that we stored in those NADHs and FADH2s. So we're going to take that energy, they're just holding it, and we're going to make ATP from that energy. But we're going to do this through oxidative phosphorylation. So it's going to take steps to release the energy and finally use it to make the ATP. And this is going to occur in the electron transport chain. The name tells you right now, we're going to transport the electrons, they're the energy, through this chain. We're going to pass it from one molecule to the next. Therefore, we're passing the energy from one molecule to the next until finally the last structure will be able to do something with that energy. Okay, this is going to occur in the mitochondria also, but it's going to occur on the inner membrane of the mitochondria. It has folds called cristia, and embedded in that inner membrane are the enzymes that we are going to need for this process. ATP is generated by oxidative phosphorylation. The final electron acceptor is going to be oxygen, that inorganic molecule. Okay, and when we take that oxygen and put it with hydrogens, we're going to make some more water, and again as a waste product. The NADH and FADH2 are going to pass their electrons to a series of electron carrier molecules, and each step carries out a redox reaction. And eventually this will release the energy to make ATP through the chemoosmotic mechanism. So this process is called chemoosmosis or chemoosmotic mechanism. Now I'm going to go into this in more detail about what's really going to happen. Now we're going to start, think of this as steps. There's 3, you can see on the figure there are 3 big molecules. Think of those as 3 steps. And if you start at 3 steps and you start at the top, you have more energy, and when you run down, you use energy running down those steps. You have less at the bottom. We're going to take the energy and we're going to jump it from one step to the next to the next. And it will lose energy as it does that, and at the end it has no energy left, and then it will have passed the energy to other molecules. We're going to take our NADH and FADH2, and they're going to enter into the steps. We'll look at NADH. If we take NADH, now NADH is holding the energy. But what I'm going to do is I'm going to pull an electron off that is holding the energy, and it's going to bounce down those steps. And as it does, it gives off energy. Energy can't be created or destroyed. It has to go somewhere. So what that electron does with its energy is push some hydrogen ions across that inner membrane into this intermembrane space, the space between the inner and outer membrane. So now we get a buildup of hydrogen ions. So there's a big concentration in there. And if you go back to remembering about diffusion and concentration gradient, we have a lot of hydrogens inside. So they're dying to run back across that membrane to get back to equilibrium. Okay? But they can't run across it except for through one area. There's a special little door structure called ATP synthase, and this will allow the hydrogen protons to run back through. That's what H plus is, a proton. So they can run back through that door. But right now, when there's a high concentration, they're now holding the energy, because once they get released, they're going to give off the energy as they run through that door. So the NADH had the energy, then the electron had the energy, it passed it to the hydrogen protons, the H pluses. We open the door, the H pluses run through, and when they run through, they give off energy. And finally, the energy they give off is used to take ADP, add a phosphate to it, and make the ATP. So we finally are making something with that energy after we've passed it down this long chain. When that H plus runs in the door, the final electron acceptor is going to be that inorganic molecule, that oxygen, so we form H2O. Okay. Now this is called chemoosmosis, chemical diffusion, because the H pluses are diffusing back across through that door into the inner membrane space. And when it does, it is going to produce the ATP. That door's name is actually ATP synthase, and when you add ASE to a word, it means it's an enzyme. So this is an ATP synthesizing enzyme, so it produces the ATP. Now we want to look at how we ended up getting 36 ATPs from one glucose. And this is because your NADHs and your FADH2s, depending on where they're made, are worth more ATP. So looking at the conversion ratio, if the NADH is made in glycolysis, it will give rise to 2 ATPs. Gives off enough energy to make 2 ATPs. If the NADH was made in the PrEP or the citric cycle, it's a little more energy rich and gives off enough energy for 3 ATPs. And if it's an FADH2, that produces enough energy to make 2 ATPs. So looking at the summary of where we made the NADHs and FADH2s and how many we made, we have 2 NADHs in glycolysis. That will produce 4 ATPs, 2 times 2. We have 2 NADHs from the PrEP. That will produce 2 times 3 ATPs, so that's 6. 6 NADHs from the citric acid cycle, so 6 times 3 is 18. And 2 FADH2s, they're each worth 2 ATPs, so 2 times 2 is 4. You add that all up, you have a total of 32 ATPs that we got from our electron carriers. Remember, we made 2 ATPs directly in glycolysis and 2 more in the citric acid cycle, so the total is 36 ATPs from one glucose molecule. This is assuming you're healthy and everything is working as best it can. In your cardiac muscle and liver cells, there's a little bit more energy that's yielded from the NADH from glycolysis. It will make 3 ATPs per NADH, so you will get 38 ATPs from one glucose molecule. So far we've talked just about the process of breaking down glucose, but there are other processes we do that may or may not produce energy, but other things and other ways to store the energy or to break it down. The first is glucogenesis. This is when we synthesize glucose from non-carbohydrate precursors. Key word there is non-carbohydrate. I'm not talking about taking fructose and converting it to glucose, which are both sugars. I'm talking about taking a lactic acid molecule, which is not a carbohydrate, or a glycerol, and converting it to glucose. Glycogenesis, G-L-Y-C-O-genesis, is the formation of glycogen. Genesis means to make. So glucogenesis, we're making glucose. Glycogenesis, we're making glycogen. And we're making that from glucose and this is how we store our energy reserve. Glycogenolysis. You throw the term lysis in there, we're talking about breaking something down. So here we're going to take the glycogen and break it back down to glucose. We will do this if you ate lunch, had a big meal, you broke down your food to make it into glucose, you have too much glucose, you don't want to throw away your glucose, you want to store it as glycogen, you put it in the storage cabinet and you leave it there. Now if you miss dinner, all of a sudden your glucose level and sugar level in the blood is dropping, you got to get sugar in there. You're going to go to that glycogen and break it back down to the glucose and put the glucose back in your sugar. Back into your blood. Lipolysis is when we break down lipids. Now, lipid is an energy storage. Okay? We are going to, if you eat a lot of food, first you'll put the sugars as glycogen, and then anything left over you're going to put into your lipids. We want to break it down at times when you are dieting, and that's the whole idea of a diet. You stop eating enough, you exercise more, increase your metabolism, your demands are greater, so you use up your glucose, you use up your glycogen, and then you turn to your lipids to break them down. The lipids are triglycerides, and they will break down to glycerol and fatty acids, 3 fatty acids. The glycerol can then be converted to pyruvic acid and enter into the citric acid cycle. The fatty acids can be broken down to 2 carbon fragments and enter into the citric acid cycle. And your fatty acids, they will yield 17 ATPs per fragment, which is about 1.5 times as much ATP as you're going to get from the same size 2 carbon fragment from glucose. However, using those lipid fragments, it may make more energy, but it uses a lot more oxygen and is a very slow process. So it's not our first go-to. Lipogenesis, that's the synthesis of lipids. This is when you ate too much food and you store it. Okay? And store it as triglycerides, and then if you run out of food, you're going to turn to it and break it down. Amino acid catabolism, the breaking down of amino acids, we do only sparingly. If you are breaking down amino acids, you are in a starvation phase. Your body has broken down all the glucose, all the glycogen, all the lipids. Everything is gone, and now it's starting to break down your structural proteins. So you are starving. Okay? And it's also not even if we could do it, it's not the best thing to do, because your amino acids have an amino group, which is an NH3 group, that has to be removed by deamination. That's going to require vitamin B6. If you're starving, you probably aren't very high on vitamin B6. That amino group will make NH2, it will join with another H to make ammonia, NH3, and this is toxic to your cells. So now your liver has to work on breaking that ammonia down to urea, which is less toxic, and the kidney has to excrete it. And then the remaining carbon chains will be used for ATP production. We do not do amino acid catabolism unless we are, starving. And when I say starving, not, oh, I think I'm hungry, I'm starving. I mean you haven't eaten in weeks, starving. Nucleic acid metabolism can be done, but not all of our nucleic acids will be broken down. We never catabolize DNA for energy. That is our genetic information. RNA can be broken down into simple sugars and nitrogen bases, and the sugar can be metabolized in glycolysis. The pyrimidine bases, the single ring forms of uracil and cytosine, can also be utilized, and they can enter into the citric acid cycle. The purine bases, which are the double ring forms of adenine and guanine, they have to be deaminated, they have to be excreted as uric acid because they're a waste product. And RNA metabolism is inefficient. We usually don't do it, and we usually try to recycle those nucleotides into new nucleic acids.